how can you write articles just because you are supported? So if you really want to write article, attend these sessions. So now the structures about the study design, what Dr. Santan Gopal has just now discussed. I'm not going to discuss in detail anything about this, but you must realize that research question is the starting point of any research work, and these are the various characteristics which should be there in a research question when you are formulating a research question. And those details I will not go, but only thing that you must refine your research question whenever you are formulating it. If you have formulated your research question properly, probably your other design will be really proper. So population, in any RCT, there should be 5 components should be there: population, intervention, comparison, outcome, and time. These 5 things should be there. If you have included, which goes by the acronym PICOT, then probably your research question is really appropriate. And this I will not go because the time is short, so I will not go in detail. But just look at this. There is a research question which is in a published article in Journal of Cataract and Refractive Surgery. This means if you see this 2 sentences, that all 5 elements are there. Like in eyes having phacoemulsification, that is the population. Ringer lactate solution, which is the whatever your intervention, compared to BSS plus, means it is that is your comparison. PIC. O is the outcome measure, which is now you see the endothelial cell count. The significant difference in endothelial cell loss. And finally the time, T, PICOT, the time frame, which is in the 6 months time period. So it means you know that all 5 elements are included in this. Probably this kind of research question, if you are formulated, probably you are really in the right track. Now, I thought in given time about the statistics, what can I deliver in 12 minutes, including research question. So I thought probably the best thing is to give you some guideline how to choose a statistical test, and just to see whether whenever a statistical test is being done, you are reading a paper, any statistical test which has been performed is proper or not. So that is one help that will provide you this lecture. Second is when you are doing research yourself, which test to be done, that also will decide. It's not that you collect the data and then you go to a statistician and tell that, okay, now run into all battery of tests and whichever is showing significant, I'll report and I'll say that my result is significant. Okay. And so anything, means any research, you have to have the null hypothesis, that is presume that there is no difference between the two, and then alternate hypothesis. If it is you cannot prove null hypothesis, it means the alternate hypothesis is correct. And there are 2 types of error which are there: type 1 error, which is also known as alpha error, wrongly rejecting null hypothesis, and type 2 error, beta error, failure to reject null hypothesis. Now to understand this, the basic principle of which test is appropriate for a particular type of research question, you must know that basically there are 2 types of data. One is qualitative or categorical, and the other one is numerical or quantitative. In numerical and quantitative, you should also realize that this data can be parametric, numerical, or the parametric which follows the Gaussian curve. If it is not following a Gaussian curve, it means it is non-parametric data, or if you don't know that what is the distribution of the data set, you should presume that it is non-parametric. But there are certain tests which are done to find out whether your data set of qualitative data is parametric or not. Okay, so having this background, now you just try to understand these 5 tables, which are really very simple. And in this, first thing that you should know that what type of question you are going to answer. That is there a difference between the groups which is unpaired? Unpaired independent samples drawn from the same population. What do you mean by unpaired group? Means those 2 groups has got no connection with the, or one group measurement is not interfering the measurement of the other group. Means what do I mean by that? Suppose I am measuring certain parameters, say blood sugar. In this population, I have 2 samples. So sample A, sample B. These 2 are different samples. So these are unpaired because when I am measuring from sample A and measuring from sample B, these are not getting affected by each other's measurement. So if this kind of question we are going to answer, comparison, then this table will be applicable. Then what you should know, what type of data you have. I told you 2 types of data, numerical or categorical, that you will really, if you hear my long lectures, then probably you will realize that, but I presume that everyone knows that. So if it is numerical data, and in numerical if it is parametric data, numerical, then in that case you have to find out whether you are comparing 2 groups or more than 2 groups. If it is 2 groups, then the test is unpaired t-test. If it is more than 2 groups, ANOVA. Don't try to remember this. It's just try to understand the basic principle. Where I'll give you at the end one reference where you will find all 5 tables, and it's just 2-page article, nothing more than that. So 2 is, if it is 2 groups, unpaired t-test, more than 2, ANOVA. If anything is showing positive in more than 2 groups, then you do post hoc analysis. These are certain tests for post hoc analysis to find out which group out of the multiple groups is showing significance. If it is numerical data, non-parametric data, then if it is 2 groups, man will be more than 2 groups, Kurzweil values. Again, same thing, the post hoc analysis. If it is categorical data, not the numerical data, then 2 groups or more than 2 groups, chi-square Fisher test and the chi-square. So this just try to understand the basic principles. If you have understood this table, how to go about further, your half job is done. Now the next table, which is only difference is there a difference between the groups which are paired? Now paired means they compare more sets of observations on a single, such as time series, before, after and crossover. Suppose if I want to see, let the previous one unpaired, 2 groups, effect of one medicine, 2 medicines, 2 intervention. So group A, group B, given one medicine to A, second medicine to group B, and then take the measurements. That was unpaired. Now paired means if I want to see the effect of medicine, which is the over time series. Say before medicine, what is the intraocular pressure? Give medicine, record intraocular pressure before, give medicine, and after that, after 2 hours, 4 hours, 6 hours, whatever your wisdom says, you decide that, and that is the paired group. Means the second measurement or the third measurement is influenced by the first measurement. Understood this? So this is paired group. So if this type of group is there, then this table will be applicable. But afterwards, just follow the same principle. Numerical data or categorical data. Numerical, parametric, non-parametric. Same distribution. If it is parametric, then 2 groups paired t-test. If more than 2 groups, repeated measures ANOVA. If it is otherwise non-parametric, 2 groups Mann-Wilcoxon test, more than 2 Friedman's test. Again, if there are multiple groups, then you have to do the post hoc analysis. Now here, categorical data, 2 groups, more than 2, Mecken-Marr test or Cochrane-Q test. Now, this one, the common mistakes what people commit, means the researchers also, is when the established journals, when standard journals also find mistakes. People don't realize the difference between unpaired t-test and paired t-test. All they consider that t-test is all the same. So that is the major mistake. That if the group is paired, they don't do paired t-test, they do t-test, which is unpaired t-test. Second mistake that they do, that more than 2 groups, what they do, suppose there are 3 groups, A, B, C. They compare A with B, t-test, A with C, t-test, B with C, t-test, and then report that, which is incorrect. It should be multiple, where it's more than 2 groups, whatever test is written here, either ANOVA or repeated measures ANOVA, based on what groups you are dealing with, paired or unpaired. So that test, if you find significance in this test, then do post hoc analysis and find out which group is showing significance. Okay. And the third one is the association between 2 variables, because the research question may find out, means you may research 2 variables, whether is there an association between the 2. In that case, say numerical data, categorical data. If it is numerical, both parametric, then Pearson's correlation coefficient. And here the R value that you find out. R, it can be from -1 to +1. -0 is no significance, no association, but -1 and +1 both perfect association. Only thing, one is direct relation, other is inverse relation. And if it is non-parametric, then this test. 2 by 2, if categorical data, if 2 by 2, then risk ratio, odds ratio. If more than 2, then more than 2, then chi-square trend, logistic regression. So this, just understand again this table, try to understand. So now here, what people commit mistakes. I think I'll go to the next one, then I'll come back to this. Now the fourth question can be, is there agreement between the assessments? Such as, say there is an instrument measurement, new instrument which has been introduced into market. You want to compare with the gold standard. Something is gold standard, you try to compare that. Screening, so such as screening test, diagnostic test, or rater validation. Now this type of, if the question is there, then you have to use this table. If it is numerical data, then intra-class correlation coefficient. And if you are doing a graphic method, then Blank-Altman plot. And if it is categorical data, then this test. Now here, what people commit mistakes? Common mistakes. Suppose, say, they want to, there is one instrument which is introduced into market recording intraocular pressure, new one. Now they want to compare with, because then you have to validate this. you have to compare with gold standard. That gold standard is right now is applanation tonometer. Okay. In that applanation tonometer, they take measurements of patients in the applanation group with one instrument, the same patient with the new instruments, and then they take the mean of both the readings and then they do t-test, which is absolutely incorrect. The second thing, that they will take measurement and then they draw the Pearson's correlation coefficient. And they find out Pearson's correlation coefficient 0.9, perfect correlation, so both instruments are validated. Again, it is wrong, because time doesn't permit me to explain that why it is wrong, but that is wrong. So in that situation, this is the test, intra-class correlation coefficient, which should be done, and that is the right test. And the final one is there difference between time survival trend, because the survival graph that you have to draw, but this survival doesn't mean that death has to take place, because usually this test was introduced when for cancer patients, because in that time when the survival for 5 years or 3 years was the real success which was considered, not like now what it is today. So, but in any other field, for any event to occur, say like glaucoma, if you have performed anti-glaucoma surgery, so what is the time that you want to see the end result? That when, say suppose you are starting with 50 patients, then after how many years? So suppose 1 year, how many grafts fail? After 2 years, how many grafts fail? After 3 years, how many grafts fail? So that is the event which is occurring. So if that type of situation you are dealing with, then this is the data that you have to follow. This is the table that you have to follow. Now, after explaining all this, I told you in the beginning that don't read all this, don't try to recall and don't try to memorize this, otherwise you will get confused. This is the reference which is freely available, which is open access. So just log into www.jcor.in. Sorry, I am wrong. This is the Indian Journal of Ophthalmology. So it's Indian Journal, so www.ijor.in, and in that you find out this reference. Only 2-page editorial written by us, and in that you will find all 5 tables and little more extra explained about this, whatever I have talked. Thank you very much for your patient listening.
But I think he has become an expert so that we can guide any ophthalmologist for All India Ophthalmic Society. Thank you, Varun, for your wonderful talk, as usual. Now I invite
has published numerous articles in peer-reviewed journals, and not only that, but he is on the editorial boards of several of these peer-reviewed journals. He's an excellent teacher and lecturer. You're going to enjoy this presentation. Dr. Al-Tabi.
Thank you, Jerry. This is analysis and interpretation of clinical research data. And like several of my other talks I've given at this meeting, sort of my lessons learned in clinical research, and some of the things I've learned hopefully will be illustrative and useful for you. By way of disclosure, I have no financial interest in any of the products, which I don't plan on mentioning during this presentation. This was a study that was published in 2006 in the journal Ophthalmology. It's a very important study for those of us who do keratoprosthesis surgery, because this provided good evidence-based data to support the usefulness of the keratoprosthesis, and was what we used to get insurance companies to pay for the device in the United States. And in this paper, there were several tables. This is 2 of them here, which they looked at the preoperative visual acuity of patients and then the postoperative final visual acuity of patients, again as part of the evidence to support the utility of the Boston keratoprosthesis. And that was actually part of the study. And when you're looking at keratoprosthesis surgery, you're looking at results, you're looking at vision, complications, and retention. What they discussed was the retention rate at an average follow-up of 8.5 months was 95%. But you see the follow-up goes from 0.03 months, which is 1 day, to 24 months. So not surprisingly, the K-pros were retained in those patients who had 1 day of follow-up. The causes of failure were blah, 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 blah. They did provide a Kaplan-Meier survival curve, so we could take a look at the number of retention failures over time out to the study about 24 months. So after this came out, I decided to publish my own series of keratoprosthesis outcomes, and so this was a paper I submitted to the same journal, to Ophthalmology, entitled Boston Type 1 Keratoprosthesis, Improving Outcomes and Expanding Indications. Here's an abstract, and I reported the same things that the multi-center study did, which was final visual acuity, retention, and complications. You see here in the lower shaded area, if you can't read it all, I'll read it to you. talked about the preoperative visual acuities and an average follow-up of 14 months, range as low as 1 month. The postoperative vision was improved over the preoperative vision in 90% of eyes. So looking at the final visual acuity for these patients. Overall retention rate was 84%. Average follow-up of 14 months, range anywhere from 1 to 43 months. And discussed other subgroups as far as retention, and again provided a Kaplan-Meier survival curve. So I thought, well, if the first paper was published, then this should be published, because we have similar data analysis. This is a single surgeon series, which in many ways is more useful than a multi-center study. So this is the response I received from ophthalmology. Your manuscript has been carefully reviewed, and some revision has been recommended. Below are the reviewer's comments. If you're willing to respond with the revisions, ophthalmology will consider publication of your work. So here are the comments, and I think I want to read this to you because this is important. Apropos final visual acuity, it is best to report it at set time intervals. Please read the JABS AGO editorial on improved reporting of case series and the paper by Di Loreto on best and final vision from the archives. For other measures such as retention, reporting at set time points, of course, makes sense as well. So, for example, these data have little meaning absent some kind of time denominator. Quote, in a single surgeon series of 57 cases, Boston type 1 capro implantation in patients with unilateral and bilateral corneal opacification results in improved vision in 90% of eyes and an 84% retention rate. I'm not going to go through all of this, but suffice it to say the reviewer had a few comments. These are the general comments you see here. And then had some specific comments as well. I've enumerated these. We'll just keep going through these. And I don't know, for those of you who have published, tell me if you've ever seen quite this number of comments coming back from a single reviewer. Maybe you'll have 2, sometimes 3 reviewers. We're up to 35, up to 40, up to 45, up to 50, 55 specific comments after about 10 general comments. So, I mean, the fact that this article was published shows, I think, my stubbornness as far as responding to each and every one of these comments. But this is one of the editorials that the editor of ophthalmology referred me to. And this is the article where I dug jabs, and I think this is a very good article for any of you who do clinical research to read. And I'll read this as one section. An analysis reporting the proportion of a series of patients with variable follow-up does not adequately account for the differential follow-up among patients and is a potentially misleading way to present data. Sir? Sir? Excuse me. Can you stop talking, please? Instead, the proportion of patients at presentation or study entry and the rate during follow-up should be reported. Similarly, an analysis in which the final visual acuity of a series of patients with variable follow-up is reported does not adequately account for the effective time and may also be misleading. Reporting the rates of rates of visual acuity events during follow-up, like falling below a certain threshold of vision, 20/50 or worse, is preferred. Alternatively, when there's nearly complete follow-up, reporting the distribution of visual acuity at specified time points, like at 1 year, 2 years, etc., is appropriate. Small case series should not be overinterpreted because of the effects of chance. I thought this was actually pretty sound, and obviously we did not follow that. And the multi-center study didn't follow that, and I didn't follow that in my original submission to the journal. This is the other article that I was referred to and I read as well. In their conclusion, despite potential bias with the use of best or final visual acuity outcomes, these endpoints alone were used in most studies published in the year 2000 in the 3 most commonly cited ophthalmology journals. Authors of clinical studies should consider avoiding the use of best or final visual acuity outcomes whenever possible to minimize possible data misinterpretation. If best or final outcomes are used, authors should consider discussing limitations of these methods. So here's a diagram, or a histogram, looking at the articles published in AGO, Archives Ophthalmology, and Ophthalmology, 3 of the leading journals in our field, looking at the year 2000, and looking at the percentage of articles that used best in the clear box, final in the gray box, or interval in the light gray box, visual acuities. So they say we should be reporting interval visual acuity instead of best or final. What you'll see in each of these journals, a minority of these reports contain reporting of interval visual acuities. As a corneal specialist, I want to see how we did compared to our colleagues. We see that in our area of ophthalmology, our neuro-ophthalmology colleagues are probably the best as far as reporting interval visual acuities instead of final visual acuities are best. In cornea, we're in the middle of the packs. Pediatrics bring up the rear, looking like almost always reporting final visual acuity instead of interval visual acuity. So this is the paper that was published after responding to all those reviewer comments. Again, that was just one of the 2 reviewers. And you see I followed their advice, and I have in every publication since. So we're looking at preoperative and postoperative visual acuities, not final, not best, but at specified time points. And this allows me to compare my results to other authors who are publishing on their outcomes of Boston ketoprothesis, looking at 1 year, what percentage of patients have vision of 20/20, 20/50, etc. And also we're looking at complications and retention. So we see here, when you're talking about a rate, the mistake I made in that first paper was I talked about a percentage. The retention rate was 87%. That's not a rate. A rate takes into account a unit of time. So you have to say at 1 year, the percentage of K-pros retained was, for example. Or what we did in this case is looked at the number of ketoprotheses that had to be removed, called a retention failure rate, which was 9. And then looked at the cumulative follow-up in terms of time of all the patients who had a ketoprothesis. In that case, the cumulative follow-up was 97 years. So now you can say that the retention failure rate is 9/79, or 0.11 per person year. That's the way you should report a rate. And that's the way I can compare my results to somebody else who does ketoprotheses. That's important, because if our postoperative managements differ, we want to see what's the incidence of infectious keratitis, for example, using my antibiotic regimen versus somebody else's. You really have to look at the rate of infectious keratitis in my series compared to the rate in their series in order to say which one may be better. We're looking at just complications without taking the extent or follow-up into account. You really can't make that comparison. So you see down here, because 4 ketoprotheses were removed in the first year after surgery, the 1 year retention rate was 90%. Because 5 more were removed in the second year, the 2 year retention rate was 61%. Now I can speak about a rate, because I'm talking at a specific time period after surgery. And here we see the table of all the complications. Again, not just mentioning these, but breaking them down as far as when these complications occurred, so we can compare between studies. So what lessons did I learn from publishing what was my first publication on the Boston ketoprothesis? Many post-op complication measures, outcome measures, such as visual acuity and complications, are significantly affected by the length of follow-up. So the reporting of such measures at specified intervals after surgery removes bias associated with variable length of follow-up and allows for easier and more accurate comparisons of outcomes between different studies. The reporting of post-op complications in terms of rates, the number of events or cumulative time of follow-up, allows an accurate assessment of the event risk in a given study and permits the combining of data from different studies for, let's say, a meta-analysis. I want to thank you very much for your attention.
Thank you.
Outcome, you have to talk not the final outcome. If you have follow-up of only 1 day or 1 month, and you have a patient going up to 5 years, it makes a lot of sense to talk about this. At the various intervals, what are your success rates, the drop-off rates, or the visual outcomes, the complications. I think it makes a lot of sense.
Right, and we've done the patients with 1 day of follow-up. It should obviously be not just interval, but have a minimum length of follow-up in order to really be able to comment intelligently on complications following ketoprothesis surgery. I found one other thing I think has to be considered is that also even if you control for length of follow-up, if some complications, if the incidence of complications differs over the length of time, you can still introduce bias even if you're controlling for length of follow-up. For example, if you develop a retroprosthetic membrane, usually within a month after surgery, if you follow some patients out for 10 years, you know they won't develop this complication between 1 month and 10 years. So having that long follow-up doesn't necessarily, it's going to skew your data. So you have to, I think, realize or customize the way you analyze the data depending on what you're analyzing. In this case, if it's complications, looking at early post-op complications, and if you're looking at something that instance of which occurs over time, like infectious keratitis, making sure, again, you're controlling for the length of follow-up, because obviously these complications are going to become more common the longer you follow patients out after surgery.
However, the data can be skewed for other reasons, even with an excellent follow-up period without surgical complications, because before surgery you may not know what other pathology
1 or 1 week after a K-pro, we look and there's a retinal detachment. There may be some pigmentation showing it's chronic. So on this complications table, it will show up as a retinal detachment diagnosed more than a month out. Because then I would think, okay, it's more likely actually that it developed during the post-op period and was not pre-existing prior to paracesis implantation.
And also you have the situation of glaucoma.
And glaucoma, that's another good example.
Well, thank you. I know you have a big trip ahead of you, and thank you for all the work you've done during this session.
Thanks for inviting me to be part of this post.
No, I don't have any comments on the paper.
Of this session. So it's a double summary. I'll start with the quote from Albert Einstein, which is good for, remember, Albert Einstein was a genius, and he could think of all types of complicated matters and be able to create a clear path through confusion. And he came up with this statement of all people, make everything as simple as possible, but not simpler. So obviously he was not only a great researcher and innovator, but a good teacher today's session, and then tell you how to summarize. We have different types of articles, and all of these articles in all of the journals in the world generally follow a protocol that's set up by international standards. And this includes the Indian Journal of Ophthalmology, the American Journal of Ophthalmology, the Archives of Ophthalmology, Nature Science, New England Journal of Medicine, etc., etc., etc. In this case, I'm going to break my talk into various categories. Different types of articles that are published. Laboratory experiments that are just taking place in a lab. Next stage would be animal trials, and then clinical trials with humans. And finally, the outcome measurements. Articles, as mentioned earlier by Dr. Gupta, start with an abstract. It follows the title, and the title should be limited so that it grabs the interest of the reader. It goes to the core of the report, the purpose of the article, the hypothesis it plans to prove or disapprove, its findings, and how useful it is. And also it emphasizes the importance of these results or the lack of results. And it gives the scientific contribution and structure to the paper. That's the abstract. The introduction, as discussed by Dr. Gupta, discusses the materials and methods used for that investigation and references to similar investigations and findings. It gives a focus to the paper. It brings the reader up to date with some of the knowledge that has been distributed earlier, so that someone that's not so familiar with the topic will learn about what has developed prior to this paper being published. And the results have to present the data without interpretation. The facts, just the facts, nothing but the facts. And at the end, the discussion is more liberal, because it interprets the data from your studies and compares it with similar investigations that previously reported only in peer-reviewed journals. And finally, the summary, the conclusion. That's my topic, to summarize. And this emphasizes the significance of the report by highlighting the data derived by this investigation that may or may not support the hypothesis made at the beginning. And this summary could be a category of the abstract. Sometimes when you're reading an abstract, it breaks it into different categories, and at the end of the abstract there's a category called summary. Another area for that summary is in the discussion. Now I'll give some examples. Basic research, let's go into that. Pharmaceuticals. Okay. I'm going to concentrate mostly on the eye, ophthalmology journals, and just like the previous speakers, imparted some additional information, not only about how to write a paper, but some good tidbits about ophthalmology. In this case, we're going to go into biochemical and pharmaceutical research. And we'll talk about antibiotic sensitivity. And I highlighted the important areas. Usually, articles talk about buildup of resistance from antibiotics. But very little was done about how there could be a crossover of collateral sensitivity from genetic changes. And so this was in the abstract, using large-scale experiments with E. coli, that there's collateral sensitivity. That's the abstract. They set out what they want to do. In the summary, it talks about large-scale laboratory evolution experiments, genetic adaptation to the aminoglycosides, and whole genome sequencing in the laboratory. Please understand that this talk is tilted towards genetics and stem cells because that's what I'm interested in. Just like the previous speakers really tilted it towards subjects that they were interested in. So in this summary, it's good. It just talks about the results of their lab experiments, showing what they set out to prove. Let's go into genetics. Again, it's laboratory. And these are a paper about electrically assisted ocular gene therapy, electron transfer iontophoresis. They wanted to see in the laboratory which method is good for transinfection other than using AAV, associated adenoviruses. And so it talks about, in the summary almost, in this abstract, iontophoresis has been found efficient for small nucleic acid fragments, and electron transfer, on the other hand, is for the large one, oligonucleotides and plasmids. So in this case, the abstract, in a way, had the summary. But in the conclusion, or another summary, it says that iontophoresis is more appropriate for the delivery. It says the same thing. So in this case, the conclusion really reiterates what was in the abstract. And it goes on to saying that this I take affront to. It's a guess. They're talking in the summary. They're guessing. Well, they shouldn't put that in there as a guess because we want facts, only the facts in a summary. All right, let's go to stem cells, induced and redirected. This is isolation of amniotic stem cell lines with potential for therapy. And it talks about, in the abstract, stem cells are capable of differentiating into multiple lineages, and that cloning human lines, verified retroviral markings, etc., etc. This is what they set out to do. And in the summary, they conclude that, yes, it can be done, and in the summary, it makes a statement, I guess it's okay, in the future, banking of these stem cells may provide a convenient source for both analogous therapy in later life and matching. So we're talking about what they resulted. These are set as facts. It shouldn't be theory, and theory should only take place, opinion should only take place in the discussion. Now we're going into clinical trials, clinical trial type of papers. Collation of data, different phases, 1 to 3, of developed products prior to Food and Drug Administration approval, which has some impact around the world. So here, this is phase 1 clinical trials of recombinant human endostatin. Well, it gives the purpose in the abstract that they're injecting recombinant human endostatin intravenous to see how it does with refractory solid tumors. And in the summary, it says that there were no clinically significant toxicities. Okay, that was from their investigations. We'll go into collation of data after FDA approval. Once something is on the market, and this is putting together a mass of information, and you know that there are many drugs that have been taken off the market because of effects that were seen only after millions of people had taken the drug, which did not come out during the phase 1 to 3 trials. So anyway, here we have an abstract about lutein and xanthine and omega-3 fatty acids for age-related macular degeneration, the ARADS 2 study. And it was a question of whether adding 2 types of drugs to the ARADS form, lutein and xanthine, omega-3s, and EPA might further reduce the risk of age-related macular degeneration. Is it significant? Does it do anything? So the abstract asks, in effect, asks that question. It does not give an answer in this case. But take a look at the summary. The addition of lutein and xanthine and DHA, various fats, to the AIOC formula, either the lutein and xanthine and or the fats to the formula in the study, millions of people, did not further the risk of progressive to advanced AMD. Okay, now that's significant. It was negative. Comparison of low dosing versus high dosing showed no evidence of significant effect. And there's insufficient evidence to provide a clinical recommendation. The only thing is that the lutein can replace the beta-carotene that has some adverse effects, I believe, on the heart. So that's about the one thing that came out of that paper. So, in summary of my summary and summary of this session, I'll say that the summary should briefly highlight the important conclusions of the reported research or investigation. It emphasizes the significance of the report by highlighting the data derived from the investigation that may or may not support the hypothesis made in the introduction, and often this summary is embedded in the discussion section or could be seen in the final segment of the abstract. Thank you for your endurance, for staying for the last session of the next to the last day of the meeting.
So the summary of the session is already been provided.
Dhananjay Shukla is the section editor, and he also helps in reviewing Indian ophthalmology. He is a proud fit vitreoretinal surgeon from Arvind and now medical director and vitreoretinal consultant, Center for Sight in Ludhiana.
Thank you, sir. Thank you for the introduction, and thank you for inviting me to be part of the session. Dr. Schultz has already provided a summary of the session, so I believe this is an epilogue or a recap. So we have already seen how to make a paper and how to submit it, but what happens the day after? The day after means when the paper gets rejected. So just a minute. The presentation. So rejections are the reason most of the potential authors don't even try to publish. And if you dissect the rejections, the causes may be within the manuscript or they can be outside the manuscript. Within the manuscript, the most problems are with the novelty, that's originality, or with execution. And there, as we saw in the previous session, there might be ethical issues which invite rejection. But there can be causes outside the manuscript too. In the top journals, the competition is too stiff, and you are as good as the next paper which is getting accepted or rejected. Or our paper may not be relevant to the target audience, or sometimes we do not put ourselves in the shoes of the reviewer. So what are the causes within the manuscript? Most of us start with the writing careers with a case report. Something looks very exciting to us and we want to share with our colleagues. But they are easy to write and also easy to reject. In fact, a lot of journals have actually stopped the submission of case reports in an effort to improve their impact factor. And so now we are crowded for space a lot. You have a better chance if you write a case series, more likely if it is prospective and controlled, and the best chance is obviously if it's a randomized controlled trial. For example, if we write about a huge macular hole due to trauma, we cannot conclude anything except that traumatic macular hole can be really big. There is really no message to the reader here. It's a classic so what report ready for rejection. So, now to re-quote Einstein, if you say why the papers get rejected, you can simply say because you do not follow what has been advised so far. So I'll just walk you through some of the salient points, just to recap the salient points. So there can be problems with the abstract. Now it's the standalone summary and the most important part of the abstract. And believe me, a lot of papers do not make it beyond the abstract in terms of the section editor or the reviewer. So if you have a good abstract, it does not guarantee its acceptance. But if you have a bad abstract, it definitely guarantees rejection. So what can be the problems? The aims, methods, results and conclusions are not linked. The conclusions typically should follow from results and prove or disprove the hypothesis. So there can be launching problems. The research question itself is not stated well enough. It should be clearly visible in the last paragraph of the introduction, or the rationale of research is either not valid or not validated. So why we bother to do the study, what's new about it, and why it is important for the readers to know about this new thing. Now, methodology is the thing which dictates the actual destination of a paper. The top-ranked journals have the most rigorous requirement from the methodology, as you can see from the worksheet of Ophthalmology Journal. So it's so detailed that it's impossible to write a substandard paper. You need not submit for that. So it's obvious that if you prepare a junk food or fast food, you will be employed in a roadside restaurant, McDonald's. But if you really put effort and make a really esoteric dish, then you might be a chef at JP Residency. So sometimes our intervention or measurements are not suitable to the aim that we are targeting. And frequently there are statistical issues. Now actually a lot of journals advise the author, especially of the larger studies, to consult a professional statistician. And finally there can be ethical issues like not taking approval of the institutional review board, or not complying with Helsinki declaration, or revealing the identity of the patient, or not taking informed consent. For example, taking informed consent is a very important thing for British Journal of Ophthalmology specifically. They want actually a PDF of the patient's consent, with the submission. Results are the heart of the matter, and we have to understand that more is not necessarily the merrier. It's like having too many olives in the martini. So our salient findings may get cluttered by unrelated facts, or sometimes negative or unexpected findings or complications may not be stated, and this does not escape the eyes of the reviewer. Sometimes the table data is repeated actually, and tables are there to save space, not to repeat the data. And obviously, as Dr. Schultz just mentioned, it's not a place to comment on the data at all. Finally, discussion of the part is most difficult for the beginners. As Ahmed said, you are as good as the guy next to it in a bodybuilding show. Similarly, so you have to compare and contrast yourself with the existing literature and show what is new in this study as compared to the existing literature and how it is relevant for the reader. If it is not validated and uncomfortable studies are brushed in the carpet, it generally invites rejection. So unexpected outcomes, complications and limitations should be stated, and they should be discussed. And we must state why the paper still retains relevance in spite of the limitations. And on the other hand, we sometimes go overboard with the generalizations, speculation or conclusions, which again is a cause for rejection. Now, what are the causes external to the manuscript? One is choosing the wrong journal. Now, there are more than 100 ophthalmic journals to choose from among the index and peer-reviewed category. We generally prepare for the best, for the highest impact for a journal, but you have to be prepared for the worst. And sometimes our subject is too local for an international readership, and so we would probably better off in submitting to the local journal. Sometimes our subject is too specific, and so it should be submitted to a specialty journal rather than to the Journal of Ophthalmology journal. And sometimes we simply have to look at the nature of articles which are most frequently published in the journal. For example, British Journal of Ophthalmology really loves community ophthalmology articles. So if you have a community ophthalmology article, you have a good chance there. Finally, we should put ourselves in the shoes of the reviewer as well. Reviewers generally see whether this paper is written by a first-timer or an experienced or veteran. So they look at the diction, the style, the structure, the compliance of the references with the journal style, and also whether the styling betrays the previous rejection. One obviously tries to see whether it says something new or important, and if it does, then does the methodology fit the purpose? Are the results clear and crisp and significant? Are the conclusions support for the results? And are the limitations acceptable? Finally, a reviewer always sees, does it add to the patient care? This is what we sometimes fail to address. In spite of all this precaution, we might get rejections. We all do get rejections. So publishing is as much about perseverance as about excellence, because most high-end journals have an acceptance rate of about 25% or less. So one thing that we must do is the attitude to rejection should be different. We should take it that we have given it to a top-rated authority for a review, and we have got their comments. We can address those comments and concerns and submit to probably a lower end or lower impact better or specialty journal, as the case may be. We should remove the telltale sign that has been written for another journal. There is a controversy on whether we should disclose the rejection or not. Journals always encourage to disclose the rejection, but some of my senior colleagues, when I did the same for one article, it does not always invite a second rejection, but most people don't do that. Whatever you do, you should not put a manuscript in a dustbin easily. Remember that Hans Krebs' Nobel Prize winning paper on Krebs cycle was rejected by Nature before it got published in a small journal called Dutch Enzymologica. In fact, Krebs got a letter of apology from Nature journal after he received the Nobel Prize. Similarly, the first report of fluorescein angiography in a human eye by Novotny and Alves was rejected by AIOC, American Journal of Ophthalmology, and after several rejections got published in Circulation. They also got a letter of apology from AIOC. Do not always get that letter of apology, but as when we look at the achievers or the successful authors or the stars in any field, we look at their skill and their ability to score. But basically, we don't look at the fact that this success is built upon several layers of failure. And as Michael Jordan, one of the greatest basketball players ever, said he can accept failure, but he cannot accept not trying. So to sum up, several times the rejected papers are hastily written or not addressed to the journal to which they are targeted, and the lack of vigor is betrayed by the lack of originality in the idea. This is not tested by the literature. A slack methodology or vague outcomes or inconvenient details being brushed into the carpet. Remember, like fathering a child, a paper is conceived in excitement but groomed with patience, and even though the reviews may look ferocious to us, the papers are reviewed to be accepted. Thank you very much.
Thank you very much for a well-read presentation.
Like my one or two paper are being accepted, then I am open for the next paper also. But suppose my first few paper are not accepted, then I am gone. Then I don't want to write again and again.
No, I think that is
So very poor. Because people, those people who are writing again and again, they are writing again and again.
right.
So, but those who are not writing, they are not writing again and again. Our participation should be, we should be given, you should be told how to, you seniors, for the juniors or newcomers, entry ticket should be there. No?
No, it is there.
No, I think
Actually,
it is
how to
already-
write and publish, we are conducting 2 courses a year.